Do me a favor, visualize the letter Z. The letter Z floating in midair between me and you, right in front of your screen. You take a second to do that, and I'll do the same. Now, chances are you're able to see that letter Z floating in midair. But why is that? There's no photons catching the retina. There's no signal being transferred from the optic nerve into your brain. But yet for most of us, we can see that letter Z. That's because your vision doesn't happen here in your eye, but rather it happens right here. For centuries, we've been thinking about the eye like a camera and the brain merely as a screen or a processor. But for millions of blind patients, whether that be through glaucoma, trauma, or optic nerve damage, the camera itself is broken, but that processor, the brain, is perfectly intact, waiting to be tapped into. My name's Zhao. I'm a medical student at the Perlman School of Medicine at the University of Pennsylvania. I'm training to be an ophthalmologist. That's what I've wanted to do since coming into medical school. But over the past 2 weeks, I've been rotating on the neurosurgical service. And through my time there, I begin to understand the intersection of the eye and the brain and how these two organs allow us to truly see. Today, I'm going to be walking you through how we're hacking the biological code to stream video directly into the human mind without the use of this. This isn't entirely a new idea. You see, neurosurgeons have been trying to tap into the brain as the processing center since the 1960s. We've known for a while that if we zap a specific, distinct point on the cortex with an electrode, the patient sees a speck of light. That is what we call a phosphene. So the early logic was, if one electrode emits one dot, what happens when we use a grid? So like pixels on a jumbotron, if we want the patient to see the letter Z, the thought was, why don't we just turn on the electrodes on the jumbotron that would form the letter Z all at once? And just like that, they were hoping that the patients would see a crisp letter by connecting those electrodes in a line. So in theory, this was absolutely beautiful, but in practice, it absolutely failed. And the reason why is when they turned on multiple of these electrodes at the same time, all these phosphenes didn't stand out. They blended together. Patients couldn't see a Z from a T from a U. All they saw was an amorphous blob, a galaxy of stars with no connection. Because the brain isn't a flat LCD screen like your TV is, this seemed to be impossible. At that time, we had the idea, we had the hardware, but we just didn't have the ability to tap into the brain software. This is where the neurosurgical problem happens. You see, we tend to think of the visual cortex as a perfect one-to-one map of the outside world. But that's not what happens in reality. You see, the way the brain looks at the outside world is kind of like how we look at vision through a spoon. Now, if I look at the reflection of myself through the spoon, I do see myself, but I don't see the exact version of myself. I see myself with an enormous nose, small ears, and the edges of my head wrapped around the spoon. This is how the brain sees the outside world, and this is called cortical magnification. Now, if you look behind me at this house, this is what we would assume the visual cortex to see. However, in reality, this is what the visual cortex truly sees. Now, this image gets completely warped, and if we don't mathematically correct for that, it causes what is known as retinotopic mismatch. The patient doesn't see that perfect Z that we outlined through electrodes. The patient just sees a funhouse mirror of squiggly lines and different shapes mapped across the brain. Recently, researchers have been thinking of the brain less like a static map and more like a canvas that an artist can paint on. Take this sparkler. When held still, it's a dot in space, like a single phosphene. But the brain has something called temporal integration. It's able to hold the image for a mere fraction of the second. So when I begin spinning this faster and faster, you're not just seeing a bunch of dots in space. Rather, you're seeing a complete circle. This is your brain connecting the dots. This is what we call dynamic current steering. The idea is, instead of blasting the brain with different electrodes at different points in time, why don't we sweep and trace across the brain with electricity across those electrodes? This is how we can hack the brain's temporal integration system to draw a more coherent shape. This brings us to the 2020 Cell paper. Now, what makes this paper especially interesting isn't just the results. Rather, it's the method in which the researchers obtain these results. The researchers used a within-subject design, meaning they picked the same patient with the same exact implanted electrodes on the same exact day. The only thing that changed between the experiments was the algorithm. First, they ran the controls through an algorithm known as static stimulation. They essentially activated the electrodes simultaneously in order to form the shape of a letter. In this case, the letter Z. However, neurosurgically, this failed. What happened is, when you stimulate all of these electrodes at the exact same time, the electrical fields overlap with one another, interfering in the cortical tissue. It's a phenomenon known as spatial summation. Instead of distinct points of light, the neurons between the electrodes started firing indiscriminately. The patient didn't see a Z, but rather they saw an amorphous blob. When asked to identify the letter, the patients were essentially guessing. The accuracy hovered around 15%. Now, on the flip side, once the researchers switched the algorithm to dynamic current steering, they didn't just flash the participants the letter. They traced it instead. By tracing it, they were sweeping the current, exploiting the brain's temporal integration window. They tricked the cortex into combining a rapid sequence of dots into a coherent, fluid line. The data was remarkable. When researchers measured the correlation between the letter drawn by the computer and the letter drawn by the participant, in static trials, the correlation was essentially zero. But when you looked at trials with the dynamic current steering algorithm, that accuracy increased to 86%. We didn't just make the phosphenes brighter. We connected the dots, tricking the brain and increasing the information transferred to the cortex. I'm standing outside the Perlman School of Medicine at the University of Pennsylvania. This is where Dr. Daniel Yoshor, the senior author of that paper, also serves as the chair of neurosurgery. So why does this all matter? It matters because this procedure is a delicate marriage between two very different fields. You need the computational neuroscience in order to map out that distorted image correctly. And you need the surgical precision of neurosurgery in order to place those arrays without damaging delicate parts of the cortex. This is one of the only ways forward for patients who have lost the anterior visual pathway. Recently, in October of 2025, there's a paper published in the New England Journal of Medicine demonstrating a Prima retinal chip. This was a device that researchers were able to implant in order to restore vision to patients with macular degeneration. That's a massive win, and as a future ophthalmologist, that brings me a lot of hope. But at the same time, in patients who have lost the optic nerve, a retinal chip is a path to nowhere. In those cases, we need to go to the source. We need to go to the cortex. So where do we go from here? The device used in that Cell 2020 paper, the Orion, is a surface array. It sits on top of the brain. It's safe, it's stable, but it's limited by the two-point discrimination system of the cortex. We're able to get around 60 channels resolution, enough to navigate a room, but in order to recognize a face or read a book, that requires around 600 to 1,000 channels. This leads us to the great debate of the field right now. On one hand, we have people like Dr. Yoshor, who are working on surface arrays. They're safer, less invasive, and can last for an extended period of time. On the other side, we have intracortical penetrating arrays, like the work coming out of the Rofsima lab in Amsterdam, or commercial ventures like Neuralink. These devices penetrate through the parenchyma to get closer to the neurons in order to generate potentially thousands of channels. In 2020, the Rofsima lab published a study in Science in which they used penetrating intraneural needles to get close to the neurons in order to achieve massive resolution. They didn't just create blobs for the patients, they were able to produce high-fidelity, crisp letters. But as neurosurgeons, we know the cost of cortical penetration, gliosis, the immune response, the foreign body reaction. When you stab the cortex, the brain fights back. It encapsulates those electrodes, and the signal eventually fades. So perhaps the future isn't just about implanting more electrodes into the brain, it's about biocompatible electrodes. We're moving toward flexible polymers, floating neural dust, and bio-integrating meshes that move with the brain rather than cutting into it. The goal isn't just to stimulate the cortex, rather, it's to be a part of it. Helen Keller once said, "The only thing worse than being blind is having sight but no vision." As a future ophthalmologist, I will spend years learning about the eye, its intricacies, and every single detail. But my time here in neurosurgery has taught me that we're on the verge of something much bigger. We're moving from an age of biological repair to neural restoration. We're learning to rewrite the code. We're learning to turn the light back on for those who can't see. One light, one electrode, one sparkler at a time.